Our last presenter is Dr. Andrew Coogan, who will give an overview of studies of health effects and biomarkers of circadian disruption in experimental animal model studies of shift work or jet lag. Dr. Coogan is a professor in the Department of Psychology at the National University of Ireland, Maynooth. He is director of the Chronobiology and Sleep Research Laboratory at Maynooth University, as well as head of the Department of Psychology at the same university. His particular research interest is in how circadian clocks influence health and well-being. In particular, his work in his group focuses on circadian dysfunction, such as shift work or in light at night exposure, inflammation, and behavioral responses to such disruption.
Thank you. So, when I was researching this talk, I was surprised to find that there's already been a book on the subject. So really following on from what Joel and what Johnny were saying, I think if we talk about animal models of shift work, it's really not terribly useful or meaningful to actually talk about animal models of shift work. Shift work is a much too complex and intricate exposure. That's not to say that animal models don't have utility. I think if we break it down, we can identify possible components of the exposure of shift work that are very amenable to deployment in animal-based studies, such as the interrelated aspects of cycling, like dark cycles. So if shift workers work rotating shifts, they're obviously exposed to these types of rotating light exposures, and the related but not synonymous phenomenon of light at night exposure. Night work, so work, and I use the word night here to indicate in the normal behaviorally quiescent phase. Again, night feeding and shortened sleep. So I think all of these factors, we can very amenably deploy animal studies to elucidate their impact. I think on a mechanistic level, we can think about, again, a topic we talked about a lot yesterday, desynchronization of circadian oscillators. This is a lot easier to do in animal studies than it is to do in human studies, because we can look in the SCN, we can look in other brain oscillators, we can look in peripheral tissues. Endocrine disruption, melatonin suppression, glucocorticoid alterations in leptin and other gut hormones, the sort of catch-all phrase of homeostatic changes. And I think, maybe again my personal bias, but I think a really important mediating factor will be changes in immunity, immune function, and inflammation status. And then we can map these onto outcomes. So neurobehavioral changes, we know shift work is associated, for example, with increased prevalence of affective disorders such as major depression, cardiometabolic outcomes. We know shift work is associated with type 2 DM, obesity, cancer outcomes, and then everything else sort of tossed in there in the others box. So what does the evidence look like? Well, there's not a lot of studies out there that have specifically tried to take these shift work related factors and model them in animals. We did a small study a few years ago where we took mice and we said, well, we know from circadian theory that light is the main Zeitgeber. We know that shift workers are exposed to these rotating shifts. So what happens if we try to throw these mice into rotating shift-like schedules of light-dark? So we put animals under typical shift work type schedules, so 3 mornings, 3 earlys, 3 lates, and then we give them a couple of days off in DD. We made them forward rotating, we made them backwards rotating, or we gave them sort of alternating schedules where they work, for example, like 3 nights followed by 4 days off, then 3 nights followed, so with 12 hours inversions. We did 5 cycles of those patterns. And what we saw basically in our rotating animals, both forward and backwards rotating, is after the first cycle or so, basically they give up. And this isn't terribly surprising. They really don't try to track the cycling light-dark cycle. And this is not dissimilar to what we know the majority of shift workers do. If we look at DILMO, where we look at core body temperature, where it doesn't enchain to the shift work cycle. This is in contrast to what we saw in the alternating animals, which you can see when we've got the inversion, that they do jump around a lot more. So a sort of interesting thing as well, we saw that when we threw the animals into constant darkness to just look at free running period at the end of their shift cycles, that we saw in both the forward and backwards rotating animals, that the rhythm in DD was considerably longer, significantly longer than we would expect. You can see here is about 24 hours. And actually the rhythm in the alternating animals was significantly shorter than what we saw in the control animals. So perhaps implicating a longer term effect on SCN plasticity due to the different photic exposure histories of these animals under the shift work type schedules. At the end of the we also took the animals out. Well, we weighed them. They didn't get fat, so we didn't see any increased body weight. And actually, our alternating animals were more lean, significantly leaner than the control animals in our study. We did some very simple neurobehavioral tests. So we did this thing, that the novel object recognition test. Basically, you expose the animal to two objects, it explores them, then you take out one, you put a new one in. Usually the mice will recognize that that's a new object and will explore it more. But you can see our alternating animals don't do that. So maybe an impairment of working memory. When we threw the animals into just a open field test, the alternating animals showed significantly more activity, so they appear to be hyperactive, both in terms of the distance traveled, the velocity, and the percentage of the time spent mobile in the test. So maybe some new neurobehavioral changes in the alternating animals, which are the animals that showed that sort of push and pull on the circadian rhythms under that schedule. Other groups have shown other neurobehavioral outcomes. So this was in the shift schedule, this was more a jet lag schedule, that animals exposed to a chronic shift lag showed impairment in performance on a very simple associative learning task, the condition place preference task. And really interestingly, this was associated with a decrease in neurogenesis in the dentate gyrus, and that this decrease in neurogenesis was not dependent on glucocorticoids. So in adrenal ectomized animals, you still got the effect, which is interesting. I think Alec Davidson has already mentioned the importance of immunity and the immune system, and Alec's group published a really nice paper in 2010 showing dysregulation of immune response in shift-lagged animals. This is another nice paper from Deepak Sarkar's lab, looking where they exposed animals to rotating, either forward or backwards rotating patterns. Again, more in a sort of chronic shift lag rather than specifically trying to model shift work-like schedules. And they were particularly interested in natural killer cells in the immune system. And they showed that these shift-lagged animals showed significant alterations in clock gene expression patterns in these NK cells, that the NK cells show a natural, this spontaneous rhythm in cytotoxicity, which was lost in the shift-lag animals, that the amount of cytotoxicity overall was impaired, and that when these animals were then injected with a tumor cell line, that the shift-lagged animals were significantly more susceptible to developing, in this case, lung tumors than the control animals. So this is, I think, a really nice exposition of how the exposure may possibly via an immune-mediated pathway lead to increased susceptibility for developing of tumors, and possibly congruent with the idea that shift work as an exposure is important in a type of double hit hypothesis for cancer susceptibility. A really important question here is shortened sleep. So we know shift workers experience shortened sleep compared to day workers. We know shortened sleep has all sorts of effects in and of itself. So this is a really nice follow-up study to Alex's study from 2010, where so these animals got a weekly 6-hour phase shift, and over the course of a number of weeks. And these animals showed increased hours of wakefulness, so decreased shortened sleep. The shortened sleep was interesting because it entailed actually more REM sleep, but less non-REM sleep. And of course, since the majority of sleep is non-REM sleep, the overall effect is more wakefulness, shortened sleep. But within that, there was a change in sleep architecture. What's really interesting here is that they then challenged the animals with LPS and looked for exaggerated immune response here with IL-6. And what they found is that that the shifted animals had an exaggerated response to LPS. But animals that didn't have the shift but had enforced wakefulness, so were matched for the amount of shortened sleep, did not show this. So that the effect of the shift lag could not be explained away solely in terms of shortened sleep. And I think that's significant. Again, in terms of looking at alterations in light-dark cycles, there's a very nice paper from Bert van der Horst's group in Current Biology just in last year, where they exposed so these were P53 knock-in. These were P53 heterozygote animals in breast tissue, so they were susceptible to developing breast tumors. In these animals, they were exposed to weekly inversions of the light-dark cycle, so they had a 12-hour shift every week. You can see, following their core body temperature, that these animals re-enchained to the shift took a few days, as we would expect them to do. But I think this is significant. So these animals were constantly re-enchaining on a weekly basis, as opposed to what we see, for example, in the rotating shifts, where they basically give up and they ignore the cycling, the shifts in the light-dark cycle. The shifted animals got fat, so they had an increased body mass compared to the control animals. They also showed a increase, so the percentage of tumor-bearing animals was increased in the shifted animals versus the non-shifted animals. And the shifted animals also showed changes in sleep. So now, this was behaviorally scored sleep, so we can't say anything about the sleep architecture, but they had more sleep in the light and in the dark than the non-shifted animals. In terms of biomarkers, same group published this. Richard already alluded to the study where animals, so looking for biomarkers that are independent of time of day. So this is a big confound for biomarkers. If you've got a biomarker that shows a circadian variation, it's a lot trickier to utilize in a meaningful way. If you've got a biomarker that is expressed in a non-time dependent manner, perhaps that will have further downstream utility. So in this study, they exposed, it is a transcriptomic study, exposing animals to counterclockwise or clockwise cycling of the light-dark cycle, and picking out this receptor protein CD36 in serum as a potential biomarker that might be validated in human studies as a marker for shift work-induced circadian disruption. So it's not only the light-dark cycle, work during the normal behaviorally quiescent phase is significant. So there's a series of very nice papers from Caroline Escobar's group in Mexico, basically making animals work during different circadian phases. So the animals were put in these running wheels, and they were forced to exercise. And basically, when you do that during the animal's normally quiescent phase, what happens is that, first of all, we get a reorganization of behavior, that there's less behavior during the dark phase than in control animals. These animals get fat, so we see the significant increase in body fat and in abdominal fat. However, you can attenuate this effect on body weight if you restrict feeding to the normal dark phase, which is the phase where these animals would normally feed anyway. So you don't make food available while they're working, and you get this attenuation in the increase in body weight and the increase in abdominal fat. You also get an attenuation in glucose intolerance, as you see. So these are the night worker animals who were able to feed during their night work versus the night worker animals that were not able to feed during the night work. So by restricting food availability to the normal feeding phase, we were able to attenuate some of these effects. These night worker animals also show immune changes in that you find exacerbated responses to LPS treatment in terms of TNF, in terms of IL-6 in these night worker animals. You also find a loss of the normal gating of the immune response. So normally we have a difference between daytime and nighttime responses to LPS. That's lost in the night worker animals. And actually you can reverse those effects and attenuate those effects by restricting feeding. So if the animals can't feed during the night work, you don't see this effect. So this seems to strongly be making a link between night work, night feeding, and altered and dysregulated immune responses. Just finally, just to say this study from Linares et al. Used a similar sort of forced work paradigm, but they basically don't see any effect. But I think significantly, they don't see any reorganization between the activity patterns between night and dark. So they don't really find evidence for circadian disruption. They don't find evidence for body weight dysregulation. Actually, the night work animals are leaner than the control animals. And they looked at operant conditioning and didn't see any effects there. So perhaps this tells us a few things. So perhaps the circadian desynchrony is a key mediating process in adverse behavioral and pathophysiological responses in shift work-related exposures in animals. Desynchronization of behavioral and feeding rhythms may be of particular importance. Dysregulation of the immune response / inflammation may be important in mediating and joining up these processes. I think there's much untapped potential in the use of animal models in shift work research. We need to be much more focused. So we need to apply the same type of validity criteria than we would do if we were working on models of neuropsychiatric disorders such as face construct predictive validity. So we need to be much more structured in our approach if we are going to use animal models to meaningfully inform shift work research. Finally, I know I'm slightly over time, but I think this is going to preempt some of the questions that are going to come up. There's a lot of potential in animal models for answering some of the questions I think we need answering. Now, you will have noticed again that what we've talked about are nocturnal rodents. So, of course, it's the same true if you use diurnal rodent models. But more subtly than that, if different circadian phenotypes, so we all know you put different mice into DD and you'll get a range of free running periods. So can we model the effects of chronotype using animal models in terms of tolerance to different types of shift schedules? Chronicity of exposure. You can do a nice longitudinal study. A mouse lives two and a bit years. So it's a lot easier to look at exposure over an extended period in terms of the animal's lifespan. Sex, male, female differences. We've heard a lot of the results come from studies of nurses, which are dominated, so female specific. Background photic exposure. So shift workers don't exist in this world where the only light they see is down to their shift schedule. That is imposed on the background of the normal light exposure out there in the world, which of course varies by season, varies by country, varies by geographical location. So can we model that with animals? Exposure types. So we need to start combining light, cycling light, plus working at night. Cycling light, plus working at night, plus night feeding. So start to put these components together to mimic what we know happens in shift workers. And finally, age. Age may be very significant. So there's indications in some of the literature, epidemiological literature, that age of the first exposure to shift work may be significant, for example, in relative risk for multiple sclerosis. So at that point, I'll stop talking and I'll thank you for your appreciation, and I'll show you a picture of my university on a typical summer's day. Thank you very much.